Okay. Okay. Good morning, everyone. Before we start, if it is possible for all the persons who are present with us, and we are pleased, really, and honored to have them with us today, to deactivate your camera and microphone, just not to disturb the speaker. Thank you. At the end of this talk, you can send a short message if you have any questions to ask to our speaker, Dr. Katerina DOUKA. So Dr. Katerina DOUKA is the speaker of today, and it is with an excitement that we are pleased to welcome Dr. Katerina DOUKA to the virtual OISPP conference. Dr. DOUKA is one of the most top researchers in the world using mass spectrometry to explore and to identify more human fossils from tiny fragments in areas where it is difficult to find them, and also as a tool, as a zooarchaeological tool. Dr. DOUKA has an impressive contribution in radiometric dating. And she's a PhD holder from Oxford University and a researcher at the Max Planck Institute for the Science of Human History at Jena, Germany, and a principal investigator of the project FANDER, funded by the ERC. Dr. DOUKA is an author or co-author of more than 60 research articles and book chapters. She's inspiring archaeology, zooarchaeology, and paleoanthropology. Please join me in welcoming Dr. DOUKA. Thank you very much to be with us here, Katerina.
Thank you, Jalil. Thank you very much for this great introduction. And I hope you can hear me well.
Yes, we can.
Perfect. Thank you so much. So as my title shows, today I'm going to touch upon the work I'm doing, not on radiocarbon dating, but part of my more recent work and the use of new biomolecular tools to the prehistoric record of Eurasia. Thank you very much for the invitation, this virtual meeting. It's a great honor to be here. So my talk will mostly touch upon Eurasian archaeology, and as you will see, mostly about Denisova Cave. But with this slide here, I would like to just show you how the prehistoric record, the paleoanthropological record of Eurasia has become more entangled over the last few years with the new species, for example, like Homo lusinensis or even Homo floresiensis a few years ago. Or of course the Denisovans popping up every so often. And what is more interesting to us is, of course, that now based on genetic analysis, we know that some of these species have met and interbred, and I'm pretty sure we have many more other species or subspecies of humans who have mixed and interbred over the last, say, 500,000 years. I will talk about Denisova Cave and our work there. Mostly because it's not very well known outside the small groups of people who are working there, but also because it has particularly excited new findings over the last few years. So this is what Denisova, the valley where Denisova Cave is situated. This is how it looks like. Excuse me. Down here, you will see the base camp, and the cave is somewhere around where my cursor is. It's a fantastic position that has command over a large valley and a river. This is what the cave looks like outside. The river is just a few meters far from where this the sign is. And this is what the cave looks inside, or part of it. This is the East Gallery during excavation. So as you can see, it's not a particularly massive cave. It's got 3 different chambers or galleries, and some parts of it are not yet investigated. We know it probably extends a bit further to the south. However, we have a rather impressive Holocene stratigraphy going all the way up to the Bronze Age, and of course the Pleistocene stratigraphy is equally impressive further down where these students excavate. Well, Denisova Cave became particularly, it is a site that has been excavated since the 1980s, but it has become particularly interesting in 2010, where this little bone was found. It's a finger bone, a pinky bone, as we call it in English, and it was recognized as being human. Therefore, it was cut in half in a way that you see it here on the screen. It was cut in half. It was sent to 2 different laboratories, one of which didn't bother analyzing it because they thought, oh, well, it's some Pleistocene human, or probably not particularly interesting, and the other did. And this was the group of Svante Pääbo in Leipzig. And when they've analyzed it with Johannes Krause leading that work back in 2010, they realized that the DNA of this little bone did not fall into the genetic diversity of modern humans that we see here. It does not fall in the genetic diversity of Neanderthals that we see here, but instead it is a clade on its own, and it branches out as possibly with a common ancestor of the 3 groups as old as 1 million years ago, or about 800,000. This has been, these numbers have been revised since the first publication, but as you can see, it's a group of itself. And therefore, a new human species was recognized and made Denisova Cave a particularly important site of prehistoric archaeology. When it comes to Denisovan fossils, this is what we've got so far. These are macroscopically as well as genetically identified as human fossils, and we have a part of a parietal fragment, as well as 3 molars and the pinky bone I showed you before. On the basis of proteomic analysis, a couple of years ago, another jaw bone from China was recognized as possibly belonging to Denisovans, but I should point out that this is on the basis of proteomics and not genetic analysis, which are more detailed for sure. But all this is effectively all we've got from a human group that existed for certainly 500,000 years or so, at least since they split from the sister group Neanderthals. We have been particularly interested in dating the Denisovans and how old they were and when it particularly disappeared, given that we are using or have been using radiocarbon dating for the later part of the stratigraphic sequence of the cave. We visited several times, getting samples from throughout the 3 galleries in order to date the stratigraphy so that we date the layers where these human fossils were found. And last year we presented our results, which showed that for the fossils that we've got, I should say that Denisova Cave doesn't only contain Denisovans, it contains also Neanderthals. So we have Denisovans as likely the first inhabitants of the site, as old as possibly 200,000 years ago. And then we have more Denisovans higher up the sequence, as late as possibly 50,000 years ago, as you can see with this latest occurrences here. We also have Neanderthals appearing at the site, which is also an exciting finding, because of course Neanderthals, considered the European species, are actually in the Altai in Siberia for several. They have been for hundreds of thousands of years, as possibly as early as 120 up to 140,000 years, as Denisova 9 shows us. It is most likely that there have been several excursions of Neanderthals reaching the Altai, so not just one, because we know from other sites that we have Neanderthals around 60 to 70,000 years ago. for example, at Chagirskaya Cave. So it is an exciting place to work because several human groups coexisted and existed for the last 200,000 years. And we have it is very likely that modern humans have also visited the site, and we have material culture that we are possibly attributing to modern humans, but we certainly know that by that stage, modern humans by about 50,000 years ago were already in Siberia. However, as many of you will know, especially people in the field who excavate and find lots and lots of bones and lots of tools or debitage, how can we actually associate material culture that we find at the sites with human biology? How, for example, can we say who made these stone tools? This is a nice example of archaeological finds from Denisova Cave. We have from the lowermost layers, we have this type of flakes and cores. And as we move further up the stratigraphy, we have more elongated tools and blades or on flakes, going into more sort of microlithization as we move further up the stratigraphy. Lots of bones and bone points, bone tools, retouchers. But again, how do we know who made these? What is also very interesting is at the site, and in a way has been quite disappointing for the archaeologists and for us for several decades now, that this is what we get at Denisova Cave. This is what the bones look like. These are less than, you can see a finger here, so to get an idea of the size. The vast majority of the bones we excavate there are less than 3 centimeters long. So you cannot tell much. And between 2005 and 2013, of the 135,000 bones excavated, the vast majority, 97% of them, were not identified. So the idea we had a few years ago was, well, okay, we cannot tell morphologically what these bones are morphologically, but we can actually do this using biomolecular methodologies. We can normally, as some of you will know, when we have a bone, we can either identify it macroscopically. For example, we can say that this bone, based on its morphology, belongs to a horse, or we can extract DNA and we can say, okay, yes, it is a necus, it is a horse. We can also extract stable isotopes, collagen, and analyze its stable isotopes and say, okay, it doesn't seem to be a carnivore, or it's a herbivore and probably fits somewhere around here, possibly a large mammal or medium-sized mammal, maybe a horse, or we can use its proteins and its collagen. And this is what I will be talking about today. So collagen is the most abundant protein in the vertebrate kingdom. The study of proteins, of ancient proteins in particular, is called Polyproteomics. Is a rather emerging and fast-paced field of archaeological science. We know that modern bone contains 27% of collagen. The rest is the mineral, as we can see here, and collagen is nested around mineral plates that are forming all across the bone. While we have 28 types of collagen across the vertebrate kingdom, what we're more interested in, the vast majority of the collagen we have is collagen, type 1 collagen, and this is what we normally analyze. When we extract it, collagen looks something like that. It's in a form of a flaky form of gelatin. Some people have never seen what it looks like, so they're interested in usually in this picture. So this is collagen that we extract. Of course, the field of polyproteomics does not deal only with collagen. It deals with all the variety of other proteins that survive in the archaeological record. But in our case, collagen has been shown to be particularly interesting. And this is because a few years ago, 2009, these two people, Mike Buckley down here and Matthew Collins, his supervisor back then at the University of York, came up with an idea that by analyzing the sequence and the collagen and its peptides, they were able to taxonomically separate different animal bones. And this was at that time when it was first published. It was well, you know, it was interesting, but most of the applications was on zooarchaeological questions. For example, these researchers were interested in separating, say, goats from sheep, a big question in the Neolithic, for example, or different species of rats and mice and so forth and so on. The method is very simple in its concept and its application, in a way, from the bones, the small unidentified bones that we have in the thousands in archaeological sites. We extract collagen from about 20 milligrams of bone, which is a tiny sample, say about 5 by 5 millimeters. So it's a micro sample. So we extract the collagen, and this collagen, when extracted and digested and purified, it looks something like this. And this is when it's digested. So after it's been purified, we will cut it in specific known length fragments using an enzyme called trypsin. This is the enzyme we have in our stomachs to break down food. And by doing so, we're able to have known length peptides, which then are spotted and measured in a machine called moldy mass spectrometer, that in our case looks something like this. And what we get is a spectrum. Now, this doesn't mean much unless you have a reference library of known samples. For example, we need to have to build a reference library from animals that we know for sure what they are, and then we try to match them. And when we get specific peptides, and these are the sort of the signals that we get, what they look like. When we get, for example, this combination, we know that this belongs to the Hominidae, and therefore the bone fragment we've just analyzed could belong to hominin. It's generally, we cannot separate Hominidae from hominins, humans from great apes, but in the regions that I'm working, we don't have great apes, and therefore this is a particularly powerful method. The advantages of using zooms over other methods, for example, DNA, which is much more powerful, is that it's a very, very fast method. It's cheap, very cheap, and we can have we can follow a high throughput approach. So within a week, we can analyze several hundred samples, up to 1,000 samples one could do per week. So you can see that there is a lot of potential in analyzing all these bags of bones that we have sitting in our museums, especially now that we can't go to the field. So the first pilot study in 2015, in which we used zooms particularly with aiming to find human remains, was done in Oxford and Manchester. It was led by Sam Brown, a student of ours in Oxford back then. And after she analyzed in total about 2,000 bones from Denisova Cave, one came up with the hominin markers, the ones that I showed you before. It's a tiny bone, completely undiagnostic, one you would never pick out as being human. You can see here it's only 24 millimeters long. Here is the chip where we cut it to get the zoom sample. But other than that, it hardly has any surfaces. It's we think it is possibly digested. It passed through possibly carnivore's stomach because it's got polishing. And other than that, it has absolutely no diagnostic features. However, when so we call it, because the moldy spectrum was characteristic of hominin, we call it Denisova 11. It was found in this gallery and from layer 12.3. And when its mitochondrial DNA was analyzed, it was found to have Neanderthal, to belong to have Neanderthal mitochondrial DNA. With radiocarbon dated, it was greater than 50,000 years ago. So we published it as a Neanderthal in 2016, and it sits somewhere here, as you can see on the phylogenetic tree, on the mitochondrial tree that our colleagues built. However, when Svante Pääbo's group and Vivian Sloan continued and analyzed its nuclear DNA, then we were in for a surprise, because it turns out that this little bone, it didn't have only Neanderthal DNA. So if it had only Neanderthal DNA, as we see in this in this picture, if it was a pure Neanderthal, it would be blue here, like the Goyer sample that we see over here. If it was only Denisova, it would be completely red. But as you can clearly see in this picture, its DNA had both in almost equal numbers, equal amount. It had both Neanderthal and Denisovan DNA, and it is now, as we know, as it's been published in 2018, it's the first and so far the only first generation hybrid of a Neanderthal mother and Denisovan father we have. So that was quite an exciting discovery back then, and we're very happy to continue this work further. And as we have, so since 2017, we under the framework, under the auspices of the European Research Council, I've been leading a project called Finder, in which we are using exactly the same methodology. We're focusing on undiagnostic bone fragments from several sites across 2-3 regions. Now we have Central Asia and China as well, in which we first apply zooms, and if we find hominin bones, then we do all sorts of other analysis to identify what species they belong to, how old they are, and so on and so forth. Denisova remains the center of our work, because we know that it has a lot of material. It has excellent preservation of collagen, but also it has hundreds of thousands of fragments of bones that are there to be analyzed. We're also interested in other parts of the world for exactly the same questions, trying to link archaeology with the paleoanthropological record. So far, we have analyzed over 13,000 bones in the 3 years of the project. And at Denisova, we have several new findings. We'll just scroll through. We have found this small bone. Unfortunately, its DNA was not well preserved, so we don't know which species it belongs to. But it has a direct date. So this is interesting. It has enough collagen so that it's to radiocarbon date, but it hasn't got enough DNA to be, or endogenous DNA to be analyzed. We have more Neanderthal, Denisova 15, another small bone from 9.2. This is from the upper part of the stratigraphy of Denisova, another Neanderthal from the same layers where the hybrid was found. And as of the last few weeks, we have 4 new little bones. These are 18, 19, 20, and 21, as we call them. And so far, and I'm sort of slowly coming to an end, we have, from the Denisova Cave, we now have 17 human remains, either morphologically or genetically analyzed, 9 of which were identified using zooms. So you can see that we have more than doubled the number of known fossils from the cave with using this type of biomolecular approach. And what is also very interesting now that we can be more selective, and we can target, as we have done over the last year or so, we can target particular layers which we were interested because of their archaeology or because of their age or for all sorts of other reasons, or the preservation state. All these 4 bones that the new bones I've showed you just now, they come from layer 15. This is the lowermost archaeological layer at Denisova. The rest are either sterile or non-archaeological. And it dates to up to 220,000. Approximately the mean age is about 200,000, falling exactly at this stage 7 warming, the penultimate interglacial around here. So a very, very interesting period of human evolution. And by doing so, we are able to, and once we have the DNA results also back, we are able then to link particular human groups with the archaeology, something that we were not able to do before. And with this slide, I would like to conclude. I hope I made it, I hope it was, I showed you that polyproteomics is a good way of a good approach to start using in our investigations, particularly important for human evolution, but the same approach can be used for any type of, any age and any material, any bone from any age, so it doesn't have to be a very old bone. Zooms in particular is a rapid and reliable and rather cheap method for identifying bones to taxon and in some cases even to species. There are many assemblages of bones that they're sitting in our museums or our storage rooms and are waiting to be analyzed. So I'll be more than happy to advise and help interesting researchers with their work. And at Denisova in particularly, it has been proven to be a fantastic tool for finding new human fossils. And on average, we find a human bone every 1,000 bones of animals that we find. And of course, this is just an approximation, and every site will be different. But it is a lot of work, but I think what we also get back is very important, especially as the hybrid bone Denny showed us. And I would like to thank my team as well as my collaborators. This is the archaeologists from Denisova Cave, Anatoly Derevienko and Michael Shunkov, as well as Tom Hyam from Oxford, Svante Pavel from Leipzig, many volunteers, many pictures I haven't got them all up, could come and help us subsample our bones, as well as the team of Finder who do the analytical work. And thank you very much. Okay.
Thank you very much, Katerina, for this very interesting talk and presentation. We prepared, we presented. Thank you very much.
Oh,
so, if anyone have any question about this talk, we can, of course, we can help in translating it to French for people who, I don't know, who could or who may encounter difficulties in asking a question in English. So anyone, a question for Katerina DOUKA? Okay, can I start with a question, Katerina?
Sure.
Thank you. It's about zooms. My question here is it possible to apply this method, to other regions, such as in North Africa, where the human fossils are, I would say, rare, and with a a DNA with a very poor preservation?
Yes, that's a good question, and it always comes up. And the simple answer is yes. As I showed you, one of the little human bones from Denisova, it contains collagen, but it doesn't contain human DNA, at least the time we analyze it and with the method we've analyzed it. So one does not necessarily mean that it doesn't exist. So for example, if you analyzed it for DNA, it doesn't mean that it hasn't got collagen. So the simple question is yes, it is almost certainly that the success rate will be lower. But for example, we have now worked in several places in the tropics where collagen preservation is also supposedly poor, but we do find the more elevated we are, preservation is better. So, thinking in Morocco, some of the sites, especially the ones higher up, the cave sites, will have very good preservation. I'm certain about that. And as Jalil, you know yourself, we have already applied some of these methods in bone tools, and we had excellent results. So I think it's worth trying. It's a quick methodology. You can have results back very quickly, so you know whether it's worth doing it further. We often start with we explore the potential of an assemblage by doing 100 or 200 bones, and from then we decide whether it's worth persevering.
Thank you very much. So, any questions? Okay, can I ask another one?
Please do.
Thank you. So I'm benefiting from you now because you are here, so we can really ask.
Please. I'm very happy.
Thank you very much. So my question is about the sourcing some objects, because for example here in North Africa or in Africa in general, we have a problem with bone tools. And sometimes these bone tools, they are made on ivory, and we don't know. For example, here in North Africa, especially in Morocco, we have some in some sites, not really in a lot of sites. We have some bone tools made on ivory, and we don't know whether it is an African or an Asian ivory. So could this technique could help?
Yes. If it's an Asian or we cannot separate African and Asian elephant on the basis of collagen. So you have to go into DNA analysis. We actually, funny enough, we are talking about ivory because we've just finished a project on ivory objects. This is on modern ivory now. We are trying to work with the European authorities to see, as well as some NGOs who are interested in stopping trafficking of elephant ivory. So we have used zooms to identify first if these objects are made of ivory. You'd be surprised that some of them aren't. Some of the objects that are sold in the markets as ivory, they aren't. And that was an interesting finding from our work. We could not separate whether it is an Asian or an African ivory just on zooms, but there are ways of doing it using DNA and also very tiny amounts of sampling. So there is potential there of doing it.
Any questions? I have a fourth one, the last one.
Please, okay. I have just one question.
Okay. Sure.
Your method is destructive one. You destroy bones. Could you find a prehistorian who can give you human bones to destroy them, or?
Well, it is a twofold question and answer. The method works also non-destructively. I didn't mention this. If preservation is good, we do not have even to take a fragment out. We can simply immerse the object or the bone into a liquid for a couple of hours. It doesn't destroy, it doesn't change the appearance of the bone. And then we use the protein that has come into the solution in order to continue with zooms. So we can be completely non-destructive if preservation allows it. And in many, many cases it does. In terms of getting access, well, the good thing with zooms is that we analyze material that no one wants. The bone fragments are, you know, as any archaeologist knows, they are collected, they're put in bags and are forgotten about because you can't do anything with them. And until very recently, they were thrown away as well. You know, very often we go to sites and we say, okay, can you find where the bones are? Can you give us your bone, I'm diagnosing? And they said, no, we didn't keep them. We chuck them back to the river. We don't use them. We're not interested in keeping them. So you see that this bone scrap can actually be interesting.
Good. Okay, there is a question, or kind of question from Dr. Hisham El Osmani. He's with us. He's a Moroccan geneticist, and he's looking at, you know, human DNA. And probably, hello, Hisham, are you here? Hisham, can you hear me?
Yes,
Okay.
yes, of course.
Yes.
Hello,
Yeah, you can ask your question, Hisham.
Okay. First of all, thank you for your our presentation, and for me, it's a good tool. Using collagen, it's every time you have if you have a gold in your foot, you can't even forget. For me, and in Morocco, we have tracks of population modeling, like DNA, short-term repeat, like markers in genetics using. It's with pleasure to find this you study and your evolution in this tool. With Siad Jalil Bouzougar, we have an honor to practice your tools and have the chance to take in these tools. And thank you very much. And my question is, if the collagen likes or helps us to understanding the dynamic of South Africa, out of Africa, or in Africa, because you know it's in Morocco. I know with the Siad Bouzougar, we have lots of evidence, like ancient bones, and for me, it's every time this way to help us. And thank you very much.
So, Katerina, it's an invitation to collaborate. You see?
Yes.
That would be my honor. That would be my honor. And I love Morocco, so absolutely. Absolutely. You know, these days, we cannot rely on a single method to answer our questions. And this is because a single method cannot answer them. And it's simple. It's as simple as this. I've started as a radiocarbon specialist, and as Jalil, that's how we know each other. But radiocarbon has its limitations. It has its limitation. For example, it goes up to 50,000 years. So what do you do after that? We have to combine different approaches to be able to answer these type of questions. And similarly goes to the analysis of human remains or the discovery of human remains. One can say, well, I will excavate more sites and I will find who is doing what. But as Denisova shows us, you cannot. You can excavate for 40 years, and our Russian colleagues have been excavating every year since 1978 and 1982 annually for 6 months, up to 6 months a year. And it's only with the applications of DNA, zooms, dating, different approaches of dating, that we are all able now to put things in order and understand what is happening in Siberia and generally in northern Eurasia. So Zooms itself will not help answer the question of in or out of Africa, or the questions about timing of modern humans leaving Africa. But it can play a big role in identifying new human remains, which then are analyzed using DNA and other methodologies to build this more global understanding.
Well, thank you very much. Any other question to Dr. Katerina DOUKA? Okay, I have a last one, and I promise it will be the last one.
Feel free.
Is about again this question of sourcing objects is about the shells. Could this technique could be applied, for example, to source some shells? And I'm talking about some shell beads, for example, we found in different areas, and to see whether there were any networks between all these groups or exchanges or something like that, which is really something very interesting to know. It
is a very good question, Jalil. There are a couple of groups who are working with shells and shell proteins at the moment. I should say that the study of proteins, especially ancient proteins, is a relatively new one. So we are focusing on collagen, and therefore only bones or leather or parchment or ivory that contains collagen, because it is a much easier protein to study. It's called conservative. It means that it doesn't change through time a lot. So we don't have many different types of collagen, or what we do, but they don't change too much. With shells, the proteins of shells are a bit more complicated, and shells do not contain collagen. So we have to go into different types of shells, and then one has to build a reference library of each shell species, what proteins it contains. And therefore, when you analyze your archaeological sample, you have to match what you find with what you've known. So it's very new. I can connect you certainly to groups of people who are working on shells, and Bea De Marchi and his group in Italy, for example, the people who are working in particularly with ancient shell proteins, and certainly yes. It is something to do, to analyze. It's a good idea.
Okay, thank you very much, Katerina, for this very wonderful talk, and thank you for each and everyone to be with us here today, and see you this afternoon with another talk to be given by Dr. Marco Perizzani. It seems that it will be something about Neanderthals and Homo sapiens in Italy. Thank you very much.
Perfect. Thank you so much for your invitation.
Bye.
I just want to thank Katerina kindly accepting the organization committee's invitation, and also want to thank the nice audience for their presence and I hope to see you all again in Meknes in 2021.
Fingers crossed we can all be there.
Thank you very much.
Thank you.
Bye. Thank you. Thank you for the invitation. Nice to see you.
Bye. Bye.